In cancer clinical trials, we measure whether a drug works using a standard called response evaluation criteria in solid tumors. It's a method where radiologists measure tumors on medical imaging to determine if they're shrinking, growing, or staying stable. But the standard has a double problem. First, even among trained radiologists, there's significant discordance. Two experts looking at the same scan can reach different conclusions about tumor response. Human visual inspection is inherently subjective, but here's the bigger issue. In the real world, most oncologists and radiologists have never even heard of the criteria. So we have this absurd disconnect where approving cancer therapies based on tumor measurements that not only suffer from human error and inconsistency, but that practicing physicians don't actually use when making treatment decisions for their patients.
No oncologist, no radiologist uses RECIST. In fact, the majority of oncologists, 90%, have even heard of the RECIST criteria in the real world. And that's a disconnect in drug development, that we approve cancer therapies based on an endpoint that nobody uses in the real world. Not only nobody uses it in the real world, most haven't even heard about the RECIST criteria. The analogy I always use is as if we approved statins based on LDL and everyone used, let's say, blood pressure or the color of your eyes to administer therapy. That's what we're doing in cancer drug development.
At Project Datasphere, we are developing the Total Tumor Burden Index, or TTBI, an AI-powered model that uses machine learning to automate tumor assessments. TTBI eliminates variability in radiological evaluations while establishing a quantitative metric that bridges clinical trial endpoints with real-world oncology practice. Because when the endpoints that validate therapies don't align with the metrics that guide treatment decisions, patients bear the cost of that disconnect.